Good day, everyone. Thank you so much for joining us for our ACT-CTU clinical trial rounds. Today, we're very fortunate to hear from Dr. Brian Ward. He will talk to us about a very exciting topic, intentional infection trial participants. I'm very excited to hear more about that. And to tell you a little bit more about Dr. Ward, he's had the good fortune to receive his medical and research training at McGill, Oxford as a Rhodes Scholar, University of London, and Johns Hopkins. He is currently Professor Emeritus in the Infectious Diseases Department at McGill. He has contributed to many national and international committees related to global health and vaccines, including serving as a member and chair for 10 years on the Scientific Advisory Board for CIHR's Infection and Immunity Institute. Throughout his career, he has published over 315 peer-reviewed manuscripts. He's also worked closely with industry to develop better vaccines, culminating in the licensing of the world's first plant-made vaccine for human use in 2022, the Covifenz for SARS-CoV-2. With that, I would like to pass the mic to Dr. Ward.
Okay. So we're going to walk through a number of slides quickly, and there'll be some time at the end for questions. Oh, hold the mic up. I've been instructed. Okay. I think it's kind of funny that if you actually look up the famous medical statement, primum non nocere, which is first do no harm, which is what physicians have to say at the end of our medical training. It often comes up with this caduceus, which is actually not. It doesn't have anything to do with medicine. It has to do with mythology and commerce, which is kind of interesting because a lot of pharma is based on commerce. This is the staff of Asclepius, which is Greek for medicine and healing. And so if you see primum non nocere and the rod of caduceus, you know that somebody hasn't done their background research. Okay, I do have multiple competing interests. I do hold a position in pharma. I've been allowed to do that with permission from the university since 2017. First with Medicago, where I was the medical officer, and then Aramis Biotechnologies is the corporate successor to Medicago. Both of these companies have figured out how to make monoclonal antibodies and vaccines in plants. I'm a consultant for a number of pharma companies, both for vaccines and for immunomodulatory therapies, vaccine trials, shared awards. I don't accept speakers honoraria, and I do hold equity in Aramis Biotechnologies. I'm not a co-founder, but I'm the fourth employee for Aramis Biotechnology, and all of the employees for that company have to accept a part of their salary as equity. So I do hold equity in that company. Okay, so first question, are there compelling reasons to move from animal to human models? And my answer to that is a resounding yes. And just for those who are not familiar with the different phases of clinical trial development, right, first you have the idea over on the left, and I've put in approximate costs because many people just don't understand how much it costs to run these kinds of studies. But a basic scientist, a clinician scientist, can easily do the preclinical work. Many of us actually can collect enough money to do the phase 1 clinical trials. So in fact, a consortium of investigators, we've just done a phase 1 clinical trial for a Haemophilus influenza candidate vaccine with very little corporate involvement. But once you get to phase 2, phase 3, and then the phase 4, this is the transition phase 3 to phase 4 when you are licensed. The phase 3 is the pivotal study. You're talking about hundreds of millions of dollars, if not billions of dollars. And so as an investigator, as an academic investigator, you can get so far, but then unless you're best friends with Bill Gates or somebody else, you really need a partner to carry things on. Okay? I think if you just spent $1.2 billion to develop a vaccine that will never work in humans, and you kill thousands of mice in the process, the mouse might sit there and say, well, okay, we're even now. You spent a huge amount of money and you have nothing to show for it. Okay, but most people don't realize that we absolutely use a massive number of animals in our experimentation. So I just pulled up some recent numbers, right? 100 to 150 million animals used in biomedical research. 95% used mice, rats, birds, and fish. 52% were for basic research. 21% were procedures for regulatory purposes. And 46% involved creation or breeding of genetically altered animals. And 1% actually used endangered species or domestic animals. So we use a lot of animals in research. Okay. And so over the last few years, people have been starting to move away. And in some cases, it's actually way less expensive to do a study in children or people than it is to do a study in primates. And so if you actually look at some recent reviews, the structure of the murine and human immune system can differ considerably. Here's a picture of the mouse spleen versus the human spleen. For intranasal application, the rodent nasopharynx is wildly different from the human nasopharynx. And so really quite unreliable. And many people basically just say, look, mice lie, animals lie. You can't trust the data that you get from animals. And there are marked differences between human and immune models in terms of genetic sample size, immune experience, cost, age range, et cetera. So there is a compelling reason to move away from animals. And just as a single example, this is one of my favorite studies. It was published quite a while ago, 2013, but it basically looked at a range of gene responses, genetic responses, so transcriptional changes in response to burns, trauma, human exposure to LPS versus mouse trauma, mouse burn, mouse trauma, and mouse endotoxemia. And you look at the correlation coefficients over on the right-hand side, and you can see that, for example, the transcriptional relationship between the human burn and trauma experiences are very similar. There's a very high correlation coefficient between the genes that are up and down regulated. But if you actually look at mouse endotoxemia, mouse trauma versus mouse LPS exposure, it's basically random. And if you look at mouse trauma versus human trauma down here, you can see that the correlation coefficient is 0.05, right? So if you're studying the human transcriptional response, or you want to study the human transcriptional response to trauma, and you're using a mouse, you're really just completely misdirecting yourself. Okay? So with that in mind, a number of us have been trying to move towards human models. And recently, Scott Halpern and I got money to do the first controlled human study, challenge study with influenza in Canada, which will be starting. We'll be enrolling the first patients in February, March of this year. And this really is to establish an influenza challenge model in Canada. The challenge strain that we'll be using is a genetically susceptible Texas H3N2 from 2017. It's susceptible to all of the antivirals. We'll be testing young, healthy individuals. 32 of these individuals will be recruited and either infected or given placebo in a 2-to-1 ratio in our CHIM, our Center for Innovative Medicine at the MUHC. Dose acceleration starting at 10 to the 5th intranasally. And what we're trying to achieve is 50 to 70% mild to moderate illness in those young, healthy individuals. The other studies that we just got funded for, and this is a consortium of people I'll show you in a moment, based in Montreal, is looking at human tonsillar organoids based on Lisa Wager's protocols from UC, it's not UCSD, it's Irvine in California. We're also going to establish platforms for fine needle lymph node aspiration after vaccination and bone marrow aspiration in the context of vaccine development. So we'll be recruiting young, healthy individuals who are willing to have us take not only serum and blood, but also look at the draining lymph nodes and the bone marrow. The draining lymph node is where the action happens, right? When you're giving a vaccine in the upper arm, and the bone marrow is where long-term memory resides. And so these are much more invasive, but the information content from these samples is massive compared to what you can find in a mouse. And the initial goal and our industrial partner for this is Aramis Biotechnologies, because they have a new influenza vaccine made in plants following what Medicago did with SARS-CoV-2. But we also are working with CellCarta, another Quebec-based company, which is trying to define T cell profiles that are associated with success or failure. And they actually have a large amount of money from an AI consortium to actually use some of the data that we generate to look at T cell responses to these vaccines. Okay, here's the consortium. As I said, we're working with Dalhousie, which has the only other challenge unit, official challenge unit in Canada, and our challenge unit has 8 beds. Theirs has 12 beds. Each of these is basically a anteroom, which has a wash station. It's negative pressure compared to the corridor. Then the inpatient, like a studio apartment with its own washroom, which is negative pressure compared to the anteroom. So once somebody is infected, they stay in this area for 7 to 14 days, depending upon how quickly they clear the virus or when we start to treat them with oseltamivir. And we study them very intensively through that process. Here is the consortium of people. I lead it with Ciro Picciarello here, but we have investigators like Ellen de Callaway from Saint-Justin, Morgan Craig as well, and a number of other investigators who are doing different things with expertise in T cell biology, high content imaging, T cell biology, and so on. And these are the people who will be studying the organoids that we have collected from children undergoing standard tonsillectomy. We currently have 9. By the way, does anybody want to take a guess how many cells are in a 4-year-old's tonsils? Hundreds of millions? Want to guess? No. Up to about 2 billion cells. 2 billion cells. So we are putting away aliquots of 100 million cells, and we're putting away 5, 10, 15, 20 aliquots of these tonsillar cells that can then be used for these studies. So it's an extremely rich. We're using these are in my minus the liquid nitrogen tanks in my and they're monitored. So yes, we're not an official biobank, but we're using the research ethics permission that has been used by Bruce Mazer to collect these tissues for the last 20 years. Okay, so human challenge studies, right? You have to address the elephant in the room, because that first slide where I said, as a doctor, you make an oath, first do no harm, and now I'm actually bringing people in and I'm infecting them. And so, this is the mad scientist on the side. But the fact is, the value that we can get from these studies in a young, healthy individual who really isn't put at enormous risk is actually enormous. And so most of the people who volunteer for these studies are young, healthy individuals, many of them who are graduate students, in fact, who understand the science and want to participate and actually don't mind the idea of being locked away to play on their screens or to write their next paper in the CHIM. And the WHO has actually very recently put out a guidance for the ethical conduct of controlled human studies, because they are becoming much more popular, including respect for persons, equal respect for persons, minimization of suffering and promotion of human health, social justice and global health justice, transparency, honesty, accountability, and inclusivity. All of those things actually need to be taken into account. Okay, I'm going to go through this later, but the bottom line is that this is part of the WHO guidance, and I'm going to go through each of these boxes later and give you examples of, is it the most expedient way? Is it cost effective? Is there a critical component, and so on? But the bottom line is that the WHO has laid out guidelines that are actually quite good. Many WHO guidelines are not that good, but this one is actually quite good. And one of the things that I learned as I started to move into this area, because we've been trying to do these studies for the last decade, and we only got funding a year and a half ago, is we need to think about the risks to the participants, but also risks to third parties and communities. So for example, we're infecting somebody with H3N2 virus, and it's not a prison. They can walk out at any time, and they can either accept or not accept to stay for the whole time, or something happens in their life. where somebody gets sick and they have to leave, right? How do you deal with then sending somebody out into the community with a virus that you have given them that they can now spread to other people? So we've had to get in touch with Montreal Public Health, with hospital public health, and get everybody on board so that we have standard operating procedures for all of these contingencies that may occur. Okay, so it's actually, if somebody is leaving before they have actually cleared the virus, do we let them get into an Uber? Do we let them get onto public transit? And so on, right? So it's always lovely when somebody has done the work for you. And one of the main questions is, do you actually do harm? Do you do harm to these people who are enrolled in these studies, right? Primum non nocere. And this was systematically reviewed of human challenge studies, designs, and safety. And in fact, if you look at the adverse events, and more particularly the serious adverse events in the controlled human infection model studies between 1980 and 2020, so for the last 40 years, and you look at serious adverse events, because lots of people, right? Some of these studies are giving people malaria or Shigella or influenza. They're going to have adverse events. They're going to get sick, many of them. But it's the serious adverse events you worry about by decade. And you can see that in the participants' challenge and the total, there were 187 serious adverse events that occurred out of 10,000 participants. So a 0.2% rate. So you have to be prepared for somebody getting really sick, but it's not going to happen very often, is the bottom line. And I'll show you in one of the later slides what kinds of studies people have done that add up to these fairly large numbers of individuals challenged. Okay. Just a recap of the major points so far. The bar for CHIM study justification is high. They need to be carefully selected. Are there risks beyond the CHIM participant, him or herself? Of course, family and community. Targets need to be carefully selected. Is there an effective salvage therapy if needed? And what motivates the volunteers? So just as an example, right? So you're going to infect me with a potentially lethal virus and I get a donut, right? It's a win-win. That's not the person we're looking for to participate in our studies, right? Homer Simpson would not be a good participant. Okay, who first thought of exposing people to infectious agents? Well, the Chinese variolation, they actually fed fleas on smallpox lesions. They then let the ticks, the fleas die. They dried them up, they powdered them, and then they blew them into the nose. There was about an 8% mortality with this form of vaccination. But the Chinese recognized that you can only get smallpox once. And if smallpox has a 70 to 80% lethality rate, then an 8% lethality is actually quite acceptable when infection with smallpox is inevitable. Similarly, in the Middle East, many people actually figured out that you could also get leishmania only once. And so people took sand fleas and they let them bite on the back and buttocks of their children, because that is way better. Having an open weeping lesion for several months in a child is way better than having the open weeping lesion on your forehead when you're about to get married. Okay, so people have been doing this kind of thing for a long time. Okay, and of course, human malaria challenge was used in the 1900s, in the early 1900s, as a treatment for neurosyphilis. People figured out that high temperature, very high temperature, could actually cure some people with neurosyphilis. And so people were intentionally infected with malaria in an attempt to cure their neurosyphilis. Okay, and every time we give a live attenuated virus to a vaccine, as a vaccine, it's a chimp, right? With a very small risk. So MMR, measles, mumps, rubella, there is a tiny risk of post-infectious encephalomyelitis, about 1 in a million. Thrombocytopenia, about 1 in 30,000. And in Canada, one child dies every 2 or 3 years as a result of receiving MMR vaccine because of the thrombocytopenia, because we give the vaccines right around the time that little kids are learning how to walk. So every now and then, one of those little kids with unrecognized low platelets, because the platelets disappear for a few days. If there's particularly bad timing and that little child falls and hits his or her head, they can actually bleed into their brain and die from receiving a life-saving vaccine. Oral polio, vaccine-associated paralytic polio, yellow fever can cause severe illness and death in older individuals. So these are effectively CHIM studies where we've decided that the risks of the attenuated virus are much lower than the risks of the natural disease in the appropriate setting. Right? So yellow fever is not a problem in Canada. We don't vaccinate people. But if you're going to go on a 3-month cruise in the Amazon, then this tiny risk of severe illness and death is absolutely well worth it compared to getting yellow fever while you're in the Amazon. Okay, not to mention vaccinia, smallpox vaccine, and BCG, which cause a lot of damage in some individuals with immune problems. So attenuated strains can also be used in CHIMs, right? So we can actually vaccinate somebody against H5N1, and then we can challenge them with an attenuated H5N1 influenza virus or a live attenuated vaccine. Okay, so live attenuated H5 viruses have been generated using this virus made in Russia, so they can be used safely as challenge strains. Okay, here is, I think, one of the more interesting slides. CHIM studies over that 40-year period. It's interesting that malaria is the commonest CHIM study. Many of these studies have been done in low and middle. Influenza is next, right? Slightly more than 40 of these studies. Then rhinovirus. And then there are some really interesting ones like Shigella. Right? One of my friends, Myron Levine, actually did a Shigella challenge study in Maryland. And they almost lost one of their subjects because she had sudden onset of neurotoxicity from a Shiga toxin exposure, right? Cholera, we'll talk about that in a second. And then some parasites like Cryptosporidium parvum and even Schistosoma mansoni. And we'll talk right at the end about some of the things that people are doing in places like England and the Netherlands, where they are very, very open to these kinds of studies because they are really pushing the envelope in some of these places like Oxford and Rotterdam. But I mean, if you look at this list of organisms that have been tested in CHIM studies, it's not just benign things, right? A lot of these things are recognized to be some of the most severe disease in the world, but they're treatable, right? Which is one of the reasons why this is acceptable. Okay, so now just a series of slides. We're going to go through this fairly rapidly. If a CHIM study can be justified ethically, what's its potential value? So in some cases, a challenge study is the only feasible way to address research questions, including questions about blah, blah, blah. And in each of these cases, I've highlighted these words over here. Okay. CHIM provides uniquely robust data, i.e. early biomarkers of affection of immunity, things that you wouldn't be able to study otherwise. Okay. CHIMs are the most expedient way to select the most effective vaccine or therapeutic option, for example, in a pandemic. CHIM is the most cost-efficient way to develop a novel product requiring fewer volunteers. This is kind of shifting into the industrial area, and I'm not sure I completely agree with it, but this is what the WHO said. And CHIM is the most critical component for the development of a vaccine or therapeutic. So these last two, these are the things that industry wanted the WHO to say, because they can actually reduce the costs of developing a new vaccine substantially, but they also provide really unique data. So without a CHIM study, what do you have to do? So if a CHIM is the only way to get the data, then is there any other way to get data to look at early vaccine efficacy or therapeutic, early events in infection? Well, you can do household studies where you have somebody who's infected and then you recruit the whole family and then you study them to see who's going to get infected with transmission. You can do the famous ring studies that were done with the Ebola outbreak in 2014-15 in West Africa, which I actually have some real ethical concerns with. I actually turned down sitting on one of the research ethics boards for one of these studies because I didn't think it was all that ethical. But the idea is that you have index cases in clusters, and then you either vaccinate around them or you don't. And then you follow them to see who gets Leishmania donovani, or in this case, Ebola. And I really did not like that idea. Okay. What else can you do? Well, you have to be prepared to pounce on opportunities. And this is actually, this is me publishing myself with Tanya Watts in Toronto because I was one of the first Canadians to get H1N1 pandemic in 2009. And because I'm a lab rat, I dragged myself into the lab every couple of days, and then I came back to work. So every couple of weeks, every couple of months, and I had a series of cells and serum from myself because this was a new pandemic virus, and I knew lots of people would be interested in it. So I told people that we had these samples, and about 14 different labs. The National Microbiology Lab in Winnipeg asked for the samples. Several companies asked for the sample, and some colleagues asked for them, including Tanya Watts. And they started to write back to me and say, Brian, here's antibodies down here, and here is a significant response. And this is just 4 of the labs, but the other labs were pretty much the same. I made no antibodies because that's a significant level of antibody. So I got sick and I made no antibodies. So several of my friends said, you didn't have H1N1, but in fact, my friends are vicious human beings, and they actually insisted on doing nasopharyngeal swabs on both sides and sending them to 2 different labs. And I definitely had the virus, but I made no antibody response. But what I did make is a brilliant CD8 T cell response, expressing activation markers, expressing gamma interferon, and CD4 cells expressing gamma interferon. So I made a classic T cell response at the age of 55, but no antibodies. So here we pounced on this result or on this event, and we're able to publish something. But you can't always do that for infectious diseases because not everybody is a lab rat and has access. And by the way, I got yelled at by the dean, David Eidelman, at the time because he found out I was dragging myself into the research institute, even though he knew I had pandemic influenza. So I told him, go away, leave me alone. I'm doing it very carefully, but he was looking out for people. So for example, this is a study of 18 to 30-year-olds early in the SARS-CoV-2 outbreak, doing essentially the same thing. Okay, we now know that this is a new virus, it's a new paradigm. And so we jump on it and we study things as quickly as we can. And we look for people who are just exposed, right? Just like I was just exposed and just becoming symptomatic. Of course.
Probably like a very naive question is because a lot of these studies, you're recruiting healthy volunteers between a certain age group. They're still biased or skewed towards sampling bias is because that's not what the general population looks like. And there are many people who are geriatric or people living with immunocompromising conditions where the disease outcomes could be very different. So all the CHIM studies are very exciting, but there's still like this skewness and sampling bias.
Absolutely. You're completely correct. And you have to be aware of that. And that example that I gave of my own series of samples, because many people would say, you know, what you're looking for is antibody responses. But in fact, for an older person, I was 55 at that time. For an older person, what you're looking at is the much more expensive T cell response. And many people just don't measure those because they cost so much more than an antibody test. So yes, the risks of adverse events and serious adverse events go up exponentially as you start looking at immunocompromised individuals, older individuals, and so on. So it's the best you can do. But you still have to be ready to pounce on an opportunity when some of those other vulnerable populations become infected. So you have to do both to understand the disease. Okay, CHIM provides uniquely robust data, early biomarkers of infection and immunity. And here is an example, again, drawn from influenza. This was an early CHIM study done by Wilkinson at the NIH, where everybody was saying antibodies and CD8 T cells are what you need to protect yourself against influenza. And these guys said, well, that's actually not necessarily true. Pre-existing CD4 T cells were far better predictors of protection than either the antibody levels or the CD8 T cell levels. And in fact, this was one of the first descriptions that some CD4 T cells can actually act like cytotoxic T lymphocytes. So these were T cells that had different functionality from what had been described before. Okay. This is a multi-center controlled human infection study, again, of influenza pandemic. And what they were looking for were correlates of immunity, in this case, antibody titers. And what they found was that if your titer before you challenged was above this level, if you combine this blue line and this purple line, you could be PCR positive or PCR negative, but in fact, you were asymptomatic compared to those who were below this threshold, who all became positive by PCR and also had symptoms. So they were able to say, at this level of antibodies, you may be PCR positive. You may actually be shedding some virus, but you won't be symptomatic. Okay, so that's a very robust demonstration of protection that could only be demonstrated with this kind of study. And similarly, here's an influenza reinfection. This is a sequential human challenge. So they actually had H1N1 pandemic challenge a year apart, and only 7 participants who participated in the first challenge came back and were challenged again with the same virus, right? Which you cannot really do in nature. And what they found, surprisingly, was that 3 out of the 7 became positive, 5 out of the 7 became symptomatic. And so, in fact, the day 0 antibody response, either to the hemagglutinin, to the neuraminidase, or to the anti-stem antibodies that everybody's quite excited about, none of those were predictive of protection. Right? And many people would have said prior to this that any one of these things would be protective. Right? So again, the CHIM is just saying, depending upon how much virus you're exposed to, they got 10 to the 7th virus, which is if somebody like is really shedding virus, coughs in your face, or sits beside you in an airplane. So this is a whopping dose of virus. But the fact is these people were not protected. Okay, and then recently there's been some very nice studies done by Galit Alters. Galit Alter, who was at Harvard, then went to Moderna, is now back at Harvard, apparently. She didn't like the corporate life, other than the fact that it paid her a lot of money. And if you look at people with respiratory syncytial virus vaccination, and then you look at those who are protected versus those who are not protected, you can see that the diversity of the antibodies, right? You have antibodies here in terms of the different isotypes, the Fc receptor binding, Fc functions, neutralizing titers. They're making antibodies. But in fact, you are much more likely to be protected if you have a diverse array of antibodies as opposed to just one kind of antibody. Okay, CHIM is the most expedient way. CHIM is the most cost-efficient way. Okay, these are starting to move into some of the corporate interests. Vaccine efficacy in adults with respiratory syncytial virus. Here's the vaccine after, here's placebo recipients, and then they were challenged with RSV. And you can see that the vaccine is really quite effective. More caveats, right? They exposed these people 28 days after the vaccination. So you're giving the vaccine its best opportunity. You're not looking how durable the immune response is. You're just seeing, is that antibody and other cellular response that's initiated by that first vaccination, is it protective at least for a couple of weeks after vaccination? And in fact, it is. It's not 100% effective, but it's effective. This is the kind of study early in the development of an RSV vaccine where a company could say, okay, this is worth $300 million for us to invest in, right? This is now, human challenge studies are not just for vaccines. That's my interest, but they're also looking at antimicrobials. And this is a study looking at a novel, too long to pronounce drug for malaria, right? A KAF156. And what they showed was that above a certain dose, all doses above 100 milligrams were 100% effective at treating Plasmodium falciparum, the most dangerous kind of malaria. Again, this is the kind of data when you're developing a drug that can make a massive difference to your decision to go or no go for that drug. And here's a really good example of a no-go. Here is an H3N2 challenge study looking at a CD8 T cell mediated immune response. So this was a vaccine that was designed to make that cytotoxic T lymphocyte CD8 T cell response, and it was predicted to be highly effective. But in a challenge study, there was absolutely no difference in either the PCR viral load or the overall symptom score. And so this challenge study killed this vaccine. The company said, okay, it's not worth continuing with it. And so these are robust data that actually make you stop developing a vaccine or a drug. Okay, finally, CHIM is a critical component for the development of a vaccine or a therapeutic. And in fact, this is the poster child for CHIM studies, because the oral cholera vaccine was licensed almost exclusively on the basis of challenge studies. It's the first vaccine of its kind that received licensure in the United States, in Europe, and in Canada because it protected people in challenge studies. Right? And Vaxchora is available in Canada. 2016, it was 2020, I believe, in Canada that it was licensed. But this is a landmark for challenge studies, right? Because now regulators are saying, we believe these data. And this is just, does a CHIM study predict real world efficacy, and do the trials contribute to licensure? Well, I've just said for cholera, they were critical to licensure. But for several other vaccines, like the live attenuated smallpox vaccine, ACAM2000, it served both as a comparator and a challenge. So you vaccinate with the vaccine, and then you challenge with the vaccine to see if, in fact, the challenge takes in the presence of the immunity generated by the vaccine. And that was also how that same vaccine was demonstrated to be effective against monkeypox in primates, as it turns out. But the fact is that you could have done that in human beings as well. But exposing somebody to monkeypox is a little bit aggressive, and nobody was willing to do that. Okay. And while CHIMs typically involve infecting somebody in-house, so you bring them into your controlled space, like our challenge study at the MUHC, you can actually do these kinds of studies in different places. And so malaria studies are now, and for a long time actually, have been giving people malaria and then sending them out, and then having them come back every day to see if they're developing a fever. But we have tools now, right? And when I was at Hopkins as a fellow, I was actually involved in the monitoring of one of these malaria vaccine studies. And we sent people out with a heart rate monitor, and it monitored their temperature as well. And what we found was that these are young, healthy adults, and they do things that young, healthy adults do. And one of the things that increases your heart rate and your temperature is sex. And so we were routinely interrupting our study volunteers who had partners because their heart rate was going up and their temperature was going up, and we thought they were becoming malaria infected. So they had to actually send us a note saying, if something happens tonight, leave us alone. Okay, but the fact is that by using heart monitors and these other wearables, we're actually able to send people out and do these studies in a much more sophisticated way. So there's a small group here. What's your answer to this question? Is it possible to have fun in a CHIM study, aside from the fact that you're going to be paid $3,000 or $4,000 for your participation in this study? So do you think it can be fun to participate in these studies? Okay. Yeah. So in fact, I just put this slide in because I love it. Dr. David Milton at University of Maryland, he rented the entire floor of one of the downtown Baltimore hotels, which is not included in the publication for obvious reasons, because the hotel didn't want people to know that the fourth floor was filled with people who were infected with influenza. And what he did was he actually vaccinated some infected some and left some without vaccination with placebo vaccination. And then he had them do structured activities that were things like playing chess, doing calisthenics. You know that game Twister where you actually are like folding around each other on the floor, right? They did all of that. And he was able to actually study both vaccine-induced protection, the efficacy of the vaccine, and also transmission kinetics, right? Because he had uninfected people on the same floor participating in these studies. And in addition, you got a flight to Baltimore and $3,500 US for participating. So yes, indeed, it can be fun, even with influenza, to participate in one of these studies. Okay, and finally, the last two or three slides, is that there's an organization in the UK based around Imperial College London and Oxford, which are some of the most aggressive places for doing these studies. And it's called the Human Infection Challenge Vaccine Development Network, or HICVAC. And they are very interested, not just in challenge studies in wealthy countries, but they support many challenge studies in low and middle income countries. One of their raison d'etre is to actually promote this science in low and middle income countries with investigators from those countries. And so they, in fact, give out grants to look at vaccines and interventions against global impact pathogens. It's based out of Imperial College, but representatives of Oxford and other UK universities, as well as the Netherlands. Currently 548, actually, I'm the 548th member from over 30 different countries, including 15 low and middle income countries. Members of universities, industry, government, and the not-for-profit sectors are all part of this. So if you're interested, they hold a meeting once a year, and you can learn a great deal. I just went to the most recent meeting at Oxford and gave a talk. But one of my favorite children's books is If You Give a Mouse a Cookie. And the storyline of this is if you give a mouse a cookie, he'll ask for a glass of milk. And if you give a mouse a glass of milk, he'll ask for a piece of pie. And if you give him the pie, he'll ask for this, he'll ask for that. And so human studies are following a little bit along the same way, right? So that if you let investigators do malaria studies, if you let them do influenza studies, if you let them do Shigella studies, what are they going to ask to do next, right? And so, and how aggressive should you be? So there's at least one study of challenge where the volunteer said, yes, you can do 6 serial fine needle lymph node biopsies and 4 bone marrow aspirations during the course of a 12-month study. Okay, that is a lot to ask of a volunteer. But they said yes. And as I said, the average educational status of volunteers in these studies is master's degree or higher. And in fact, as part of HICVAC, there is an organization of, they call themselves CHIM survivors. They're participants in CHIM studies, and they actually are actively engaged through social networks with other people who are in CHIM studies. There are many people in this group who actually have had AEs. And in fact, one of the things that we will be doing is we will putting our CHIM participants in contact with this group of young volunteers, because it is psychologically difficult, right? To be alone in a room and now I'm getting sick, right? It is, even for a really well-balanced, mentally healthy individual, that's not a happy environment to be in necessarily, even though you think you're going to get better in the next few days. Okay. And at this meeting in Oxford, at the end of October, they were presenting on CHIM studies that even made me kind of cringe. So there is an ongoing study where they actually put a pad on the arm of the volunteer and they put Borrelia infected ticks on the arm of that individual, and they let them bite for a 72-hour period. So they're intentionally exposing them to Lyme disease. There is a group that's trying to get a hepatitis C virus, right? Because hepatitis C is now treatable, at least some genotypes are treatable. And a gonorrhea trial, right? How do you explain that to your boyfriend or girlfriend? Yeah, I'm going to be positive for gonorrhea, but it's okay. I'm going to earn $5,000. but that's just the way it is. Maybe you'll rethink your partner. Okay. If you're interested in public health, and this is the group, we're looking for men aged 18 to 35, residing in the UK, who are willing to participate in gonorrhea vaccine study. Okay, so these are on the edge, but they're going to happen, and I think they're fantastic. But so we're doing the first influenza study in Canada. What other CHIM studies? Well, now that we're starting this, we may do the first pertussis study was done by Scott Halpern at Dalhousie last year with Mayel Sharif, one of his graduate students. We've been asked by Shuxing to participate with them in doing a BCG challenge study as a surrogate for tuberculosis with McMaster. With UBC, Manish Sadarangani has just asked if we would help them with a Klebsiella pneumonia study, which is a LEN on the edge. That makes me a little bit nervous. And there's an aerosol SARS-CoV-2 vaccine. So it's a live attenuated SARS-CoV-2 vaccine being developed by Matt Miller at McMaster with Fiona Smale. And we might actually participate in that. So once you get going, right, if you give a mouse a cookie, we will be doing more of these studies. So thank you for your attention. And if there are questions, I'm happy to take them. And I put up all of the very quizzical young faces that I could think of, because this is, in fact, this is on the edge, but it is being done, and the value of the science is extraordinary. You just have to choose your participants really, really carefully and make sure they're doing it for the right reason. Yes, we will pay them well. But in fact, that's not enough, right? If somebody's doing it for the money, we do not want them as a participant in our study. We want them to be interested in the science. Thanks very much.
Very interesting. Thanks. Excellent and very interesting talk. So I have many questions, but I'll restrict myself to a couple. One is, how is this received by our REB? Like, were they cognizant of all the work that's been done from the WHO and from the UK? Like, did you meet a ton of resistance, or were they open? And the second question relating to influenza, is the method of delivery, like we're in this controlled experiment, you're delivering this dose. Does it depend on how deeply you inhale? Like, is the dose delivery well understood and well controlled, or is it a bit sort of unbreakable?
Yeah, those are really good questions. The dose delivery, we're not the first people to be using this NIH H3N2 virus, and other studies have delivered influenza using an aerosol adapter to a 1 ml syringe. And so what you do is you create an aerosol right in the nose. And so you are infecting the upper respiratory tract. And then beyond that, I mean, we hope that most people are going to be restricted to upper respiratory tract symptoms and maybe a little bit of lower respiratory tract. But once influenza is established in your upper respiratory tract, it will go down. But hopefully, it's a foot race between the virus and your immune system. And hopefully at the doses that we're giving, the immune system will be catching up as it's moving down into the lungs. So it's a dose acceleration study, and we're following in the footsteps of what other groups have done. There are 3 other studies that have used this virus. So we're starting with a 10 to the 5th dose that we don't think will achieve our 50 to 70% mild to moderate illness. We think it might give us 20 or 30%. We think the 10 to the 6th dose will get us up into that range. And we're using the same kind of aerosol distribution system. Your first question is much more interesting. Yes. So Health Canada and the FDA and the REB all feel the same way about it. So when we got this grant, I actually went to Dr. Marie Hertel and asked her if we could have a meeting and I could explain to her what we were thinking of doing. And when I explained to her what we were thinking of doing, she was actually completely open and receptive to the idea. She said, let's meet with the leaders of our REB to make sure they understand all of this, so it's not just a protocol that comes out of the blue. So in fact, we started with the head, and then we talked to the leadership of the REBs, and it was just a really open discussion about, what was required, what we should be doing. They had a number of conditions, right? They said you must have buy-in from MUHC infection control, right? So we had meetings with the head of MUHC infection control. She was on board. You must have buy-in from Montreal Public Health. And so we had a meeting with the leader of Montreal Public Health. And one of her questions was, are you recruiting only from Montreal? What if you recruit from somebody from Laval? And so she has agreed to help us in the event that we have somebody recruited from Laval who decides to leave the study prematurely. She will help navigate to get to the Département de Santé Publique of Laval. So we've walked very carefully towards this study over the past year. And that has also included interactions with BRDD, the Canadian regulator, the equivalent of FDA, with our pre-CTA meeting where we told them our plans, what we were doing. We got a lot of input from them. And they have inspected Dalhousie, and our inspection is coming in January 25th. So they are treating it in a very different way from another study or other studies where they simply say yes or no or tell you what to do. They're saying tentatively yes, but we want to come and inspect how you're going to do it. And that means where the virus is going to be stored, how it would be transported from the pharmacy. And by the way, the pharmacy, when we told them about this, they said, that's not going to happen because it's not a drug, it's not a vaccine. It's a risk to them. You are not going to do the dilution of this virus in our facility. And so, in fact, we had to buy a BSL-2 hood that we have put into one of the challenge rooms in the SIM, in order to bring the virus in a sealed vial up to the CHIM where it will be diluted in a BSL-2 hood by a virologist and then given to the nurse or the doctor to deliver. So it's been a real learning process and open, right? That was one of the first things on the WHO slide was transparency. Nobody wants to be surprised that you're infecting people, right? That's not a good headline in La Presse or if anybody still reads the Gazette, right? Yeah.
The first question, thanks a lot for the presentation, good one. For how long do you follow up the participant after infecting them?
6 months.
6 months, that's all.
That's all. Yeah. And what we're doing there is we're looking for the duration of the antibody and cellular response. Mm-hmm. But like natural influenza, we anticipate that it will be gone in most people who become infected within 5 to 7 days. So we're doing PCR daily. And if somebody is still positive at 7 days, we treat them with oseltamivir. And we know that this virus is susceptible to all of the antivirals. And they have to have 2 negative PCRs on sequential days in order to leave the hospital.
You are not anticipating that they may have some other complication beyond the 6 months related to the study?
Well, I mean, influenza infects 10% of the world every year. And so we're not really anticipating that there will be complications. There is a chance, right? As I said, there's a serious adverse event rate that happens at about 0.2%. So it is possible that we will be phenomenally unlucky, and one of our young, healthy individuals will have what's called ARDS, acute respiratory distress syndrome, from cytokine storm, right? The sudden release of cytokines in the lungs. And in the event that that happens, that individual would have to be admitted to our ICU, right? That's hope. I mean, we really hope, we touch wood, that that doesn't happen, but you have to accept that it might. And that's why, right, the head of medicine, right, Mark Roger, and the head of microbiology and infectious diseases, Marcel Baer, and the head of infection control, right, they all had to be on board and say, yes, you can do this study.
Okay. The last question, if you try to compare using this model and using animals that have similar genetic material to human, when you compare the two, is this still cost effective?
And but the only animals that have a similar genetic makeup to humans are great apes. And nobody's going to let you do this study in chimpanzees or gorillas. So the next step down would be old world primates, like rhesus macaques. And we share a lot of genetic responses. Our immune systems are very similar. They do not have influenza in the same way we do. We could do this study in pigs because pigs do, in fact, get influenza, and pigs have many of the same symptoms that humans have. But if you're infecting a pig, you have to have a BSL-3, well, depending upon what you're infecting it with, you have to have a BSL-3 or BSL-4 environment. And that's, well, the BSL-4 environment is far more expensive than a chim. So it's Vito in Saskatoon, right? So in terms of what's the right animal model, right? For example, we could probably do with a new vaccine for measles, right? Because measles, I showed you that the attenuated virus vaccine that we use now does very rarely cause some problems. We can probably make a better vaccine for measles, but what do you study it in? Because monkeys, old world monkeys don't get sick with measles, and new world monkeys die with measles. So what's the right animal model? Well, the right animal model is a human being. But pretty much everybody over a certain age is completely immune to measles, unless you go to Texas, where lots of people say, I don't want to be vaccinated. Or to Mennonite communities in Canada, where lots of people are unvaccinated, which is why we just lost our measles elimination status in Canada. But those individuals are unlikely to participate in a vaccine challenge study. Right? Okay.
So with that, I want to conclude today's session. Thank you so much to Dr. Ward for a fantastic presentation. And so happy holidays to everyone. We'll see you again in January. And please remember to sign up for our clinical trial symposium on February 17th. Hopefully you got the emails. Have a great day.